Okay, so this is the Wednesday evening session. I think that we're going to keep this session a little bit quicker. But if you want a full length discussion through the study material for this week, I recommend the morning sessions recording. But this session will give kind of a more condensed walkthrough of everything that's important and where we should be. And as you can see already from the very beginning of the first slide, we're of course living at the week 11 of the semester, even though we were supposed to cover this topic at week 10, but for reasons that we'll discuss in a few moments' time, we extended the spring break by another week. So this is the first week back from the spring break. And as you can also see, the morning session, I had to reschedule it from 10 a.m. to the noon. So that's the other change that we had for today. But this evening session at the normal time, I will provide a recording for this as well. But like I said, I'm trying to keep this a little bit more condensed than the morning session that I also have available for you as a recording. And the morning session goes in a little more detail. So I give you a little variation of what you can choose to see. Moving on, just a couple of things that I really want to talk about. I want to talk a little about this Maricopa district-wide systems outage, and especially in terms of how that's affecting our class and how that will affect our classes options, what we can do, and so on. And of course, I want to spend a little bit of time speaking about this week's chapter, which is the respiratory system. Like I said, I provide a very thorough walkthrough of all that in our full recording from this morning. So you can choose a little bit based on your level of comfort that which one you prefer to watch. As I'm certain you all have heard at this point, Maricopa Community College's system went through an IT outage, and I think that IT outage started on the 16th, so right during our spring break. And although there's always been, and I won't go into details what caused it, it's not really my domain, and I don't have all the details. You know as much as I know, but although the key goal has always been to resolve that very quickly. It has also been very important that the safety and security protocols were never compromised just for the sake of getting the system up. So for that reason, the college and leadership of the college made a decision that we took 1 week during which all of the work that needs to go into the place to make sure that the security protocols, safety of the information are ensured. So that extra week was spent for that. And as a result, of course, I'm sure, heard that the spring break was extended by 1 week. And as a result, also the spring semester has been extended by 1 week. So what does this mean for you, and what does this mean for us? Before I mention about that, I just want to give heads up that all of the critical operating systems have been restored as of Tuesday. Email, Google Tools, both employee and student portal, and internet, everything's running as before. Classes resumed at the beginning of this week on Monday, which was the 29th, I believe. And commitment that I personally always do, but also college has made, is that no student will be losing out. Your grades will not be impacted as a result of this outage. So what have we done, and how are we going to deal with this? Well, first of all the due dates on this course have been moved forward by 1 week. So we are going through material that was meant to be covered last week. We're going through it this week. So the due dates that we would have had normally set for last week are now due at the end of this week. In addition to the last day of the semester, has been pushed back by a week. And I am perfectly aware that that might generate for some students a little bit of a challenge if you have had plans for travel, work, or something else, that as soon as semester is over, you're going to head to those plans. So for that reason, I will be very facilitating for making sure that you are able to work ahead if you want to get all your work in even before the semester ends. But we will proceed with the schedule assuming that the semester runs 1 week behind where we were initially scheduled to be, and we will continue 1 week longer. But if for some reason any of you has to finish earlier, I'm gonna still have to figure out how I'm gonna do that. Are the recordings that I have available enough already, or how we're tackling it? But there will be a way that you can submit your work early. You can get this course finished when it was originally scheduled to finish, but I offer the chance that we can take the week longer if you need that. One thing that I do really want to highlight and emphasize to all of you, that at no point has any student record student information been compromised. So that has been always the priority. And as far as I know, and as far as I've heard, there's never been a risk of that. So these have been more of an additional security steps that were taken just to make sure to avoid any of them. So that takes us to talk about what I was going to talk after our spring break was going to be over. I was going to talk about the exam 2 and lab practical 2. But for obvious reasons, I had limited access to grade them since the system was down. So I do think that I actually have the grades finalized. But I still want to check a couple of things regarding those things that I would have normally checked during the spring break. So I'm hoping to have the finalized grades available for you very, very soon. And since we're living in this weird time of things being different from what we planned, remember that I am always very flexible, and now I will be especially flexible for reopening any assignments that you might have missed, extending due dates that you might need, whatever I can do to assist you. So just let me know and I will work with you. And the best way to let me know was through messaging on the Canvas messaging system. I know that there's a couple of messages just asking mainly about this network outage, and I'm going to respond to those probably right after this session. So what are we doing this week? This week we have assignments that are completely familiar to you in terms of the technical execution. So that will be a joy probably for many of you. All of those are completed through the McGraw-Hill, through the Connect. So you have your LearnSmart, which is the familiar one. You study through the textbook, answer questions, and if you don't know something, you can refer to the textbook. We also have the quiz, which is like any quiz about chapter content, as you can imagine. And we have the practice anatomy, where you study those structures, anatomical structures, and that prepares you to the practical as well. The difference from the previous weeks is that this week we actually have 2 labs for you to complete. So you can actually earn a little bit more points than you would on a typical week. So bear that in mind. I think that both of those labs are pretty good. It's worth working through them. So what I've listed here is the labs. So they're going to be the mechanisms of breathing and pulmonary function tests. Obviously, those due dates that were there before are not now implemented. They have been moved. All of the assignments for this week are naturally due at the end of this week. So on the Sunday evening, a minute before midnight. And I believe that that's April 4th. So I think the next step for us to is really go through the key points from this week's chapter. And to do that, I am going to move away from this presentation and open up material from our study material. So bear with me as I move from one window to another. And like I said, I'm trying to keep this session a little bit more condensed. So the full version is always on those recorded lecture videos that you find on the Canvas modules. And a little bit more extensive review is on the morning session, but this is kind of a condensed version. And now you should be able to see my screen as well. So we are going to look at the respiratory system, and we're going to go through that kind of highlighting the key anatomical points first, and then moving on to talk a little bit about the physiology. And here and there, we'll add in some pathology and other facts that are interesting and relevant for us. But really, to put everything on the map of what we have covered, where this course is heading, we have spent quite a lot of time talking about the cardiovascular system, looking at the blood, heart, and blood vessels. Well, we saw that the cardiovascular system, and especially the blood vessels, were closely connected with the lymphatic system, and immune system was then the physiological system that lived inside the lymphatic system structures. So we saw the relationship between those. But there is going to be a significant relationship also with the respiratory system and cardiovascular system, and especially the blood and blood vessels that transports the gases. Once we get gases to the lungs, then further to the rest of the body. And when we get gases from the rest of the body to the lungs, then transporting those out from our body. And in fact, when we look at some of the first responses that we expect to face when we are dealing with a patient who's unconscious and not breathing, there's three things that we're focusing on. And the first one of those is making sure that the airway is clear. There's no blockages anywhere. We want to ensure that air is able to travel to the lungs. The second one is that if the airway is clear, but the patient casualty is still not breathing, we do have to try to offer supplemental breathing. And those are those rescue breaths that you can see people doing during the CPR. And final step is the chest compressions. If the heart is not beating, we try to stimulate that. It's never going to be as efficient as the normal heart beating, but it's going to achieve at least a little bit of the blood moving within the system. So we're able to maintain some blood transporting oxygen, removing carbon dioxide to the body. So actually, this order of these priorities has changed recently a little bit in the literature, and now we are, based on current recommendations, starting that the priority is this chest compressions if there is no heartbeat on the patient. The idea is that there is some amount of oxygen still in the blood, and as long as we get that blood moving, we're already on the better side of the things. Of course, in long term, that's not sustainable. And as the second steps, ensuring the airways being clear, and then the rescue breaths. But if you feel uncomfortable giving rescue breaths, just doing the chest compressions alone is considered as a good effort and worthwhile effort. But if you look at those, out of those three things that were important things, airway and breathing, so 2 out of 3 are dedicated to the respiratory system. So respiratory system is really, really, really important for our basic survival. And this equation or formula that we're seeing here is something that you are probably familiar with from our study of the cellular respiration at your basic biology classes, Bio 181, Bio 156, whichever was your basic review of the basic introductory bio. What we see here is that we have sugar molecule that can be any type of a sugar, and we also have oxygen. Both of those are taken into the body. And as a result of them, we are able to produce energy in shape of ATP, which was the currency of the energy in the body. And as a waste product, we are left with carbon dioxide and water. So the big question here is going to be that how do we get some of that oxygen into the body, and especially not just into the body, but to those cells in the systemic circuit that need that oxygen? And how do we remove that carbon dioxide and potentially also water as well? The obvious answer to this is going to be, of course, by recruiting our respiratory system. So this is why respiratory system is so important. And if you really look at this formula, this is all that your body does. Your body does simply cellular metabolism to have that energy to perform other functions. So really the oxygen and carbon dioxide, getting some into the body and removing some becomes super important in this process. So that's why if we really start thinking about the function and purpose and how meaningful respiratory system is, we are left with the need to supply oxygen and remove carbon dioxide for the body. There are some additional functions such as sense of smell, speech, which is an important communication means for us modern humans, that also rely on structures from the respiratory system. And like we saw earlier on, there's a lot of overlap with the respiratory and circulatory systems in terms of how they achieve the overall goals for the humans and what we need. Next thing that I want to talk about a little bit is the different parts of the respiratory system. And we are going to go through these different parts, following the order of them as if the air travels into the body and then out from the body. So we'll start with the nose, nasal cavity. We'll talk about paranasal sinuses and pharynx and larynx. Then we talk about the trachea, which is the windpipe, primary and secondary bronchi, bronchioles, and branches of other branches of the respiratory tree, and finally look at the lungs and alveoli. And that completes our anatomical review of the respiratory system. Of course, air does not just travel into the lungs, it also travels away from the lungs. And sometimes some textbooks prefer to separate these structures into upper and lower respiratory system. And there's some amount of debate depending on which textbook you use, that where we draw the line between upper and lower respiratory system. So I would not consider that that's something that you have to worry about too much. So we're going to start by looking at the nose and nasal cavity. And really, if we think about how do you get air into your body, it is most of the time through the nose, through the nasal cavity. And only when the need for oxygen or need to get rid of carbon dioxide is not met through the normal breathing through the nose and nasal cavity, only then we recruit the oral cavity. But by default, it is the nose and nasal cavity. And you'll notice once we go through a material a little more that why that would make sense and why that would be so important for us. So without further ado, let's start with the nostril, the most anterior part that I can think of, the first part where the air enters. And what we notice is that there's already a filtering system in place there through those hairs that filter the large particles in terms of the air that we are breathing in. So that's going to filter some of the particles that we don't want to breathe in, but some of the smaller ones are still going to be able to enter to the nasal cavity. But we have another way of catching things there. And what we're going to be focusing on is going to be the mucous membrane. So we have skin covering the outside surfaces of our body, but inside, through the digestive tract and respiratory system, digestive tract starting from the mouth, coming out from the other end, also our urinary system, reproductive tract, and so on. We have this layer protecting those structures. It's not quite as thick, not quite as strong of a barrier as skin would be. This mucous membrane is thinner. It's very humid, sticky surface. Blood vessels are closer to the surface of the mucous membrane, and it really is characterized by the presence of goblet cells. And goblet cells are what produces this mucus, this sticky layer on top of this mucous membrane. And the functions of mucous membrane are going to make sense for us in a few moments' time. But before that, let's talk about this next function, next structure that we see here. And these are those protrusions from the lateral wall of the nasal cavity on both sides. So our conchae. Conchae refers to these protrusions that you see here on both of these diagrams. Usually they're not quite as symmetrical as we see on these diagrams from the textbooks, from the study materials, but I think it's good to start with somewhere and we can add more level of complexity later on. But as long as you appreciate that they do come out from the nasal cavity wall, you're in a good place. And you might ask me that why would you have conchae? Well, there's two functions that I can propose for you. First of all, they increase the surface area of that mucous membrane within your nasal cavity. And that makes sense because we really want to have as much of the air traveling through the nasal cavity to get in contact with that mucous membrane. So let's make more of that. Let's offer more surface area where this air can get in touch with. The other function is that the simple structure, shape of the conchae actually generates this kind of a turbulence. If you think of a twister turned on its side, on a horizontal angle, causing the air to really circulate inside the nasal cavity. Again, ensuring that as much of that air gets into contact with the mucous membrane as possible. Another structure that we see here is going to be the nasal septum. So nasal septum separates the right and left side of the nasal cavity. Usually not, again, quite as symmetrical. We have quite good symmetry on this CT scan that we see on the upper right corner. Nasal septum can get broken. We can have issues doing nose job and so on. So we would need to pay attention to that as such. But at large, in normal person, separates the right and left sides. A few other structures that relate to the nasal cavity. So here we, of course, have an individual that's looking to the left. So the anterior side is at the left, posterior at the right. And here we see the nasal cavity, mouth underneath. We see the nose with the nares, and there we would have those hair that filter the air. So if we look at the floor of the nasal cavity, which of course then would be the roof of the oral cavity, it can be divided into two parts. More anteriorly, we've got a hard palate, and hard palate is characterized by having bony structure inside this soft tissue. Posteriorly, soft palate. There's nothing but soft tissue there. There is no bone within it. And the very end of the soft palate, of course, makes the structure a uvula that we've seen before. So that's about the floor of the nasal cavity. And what I want our attention to be directed next is going to be the sinuses on this area. So if you remember the anatomical medical language, sinus was any kind of a cavity within an organ or a tissue or structure. And usually we do have a tendency of using just sinuses in layman's language to refer these paranasal sinuses, which would be the proper term for these cavity spaces within the bony structures of the skull. But there are sinuses elsewhere in the body as well. So even though you see people using sinuses, we really should specify that we're now talking about the paranasal sinuses. And there's going to be 4 that we will have a look of. And let's start with the frontal sinuses. So frontal sinuses are located right above the orbits of the eye on both sides within the frontal bone. So within that bone, there's this empty gap inside, above the eye socket on both sides. The next sinus that we'll see is going to be our maxillary sinus. Maxillary sinus located at the maxilla, the bone right underneath the eye socket, forming kind of where the very front part where our cheeks will connect is located. Another set of empty gaps. We also have a sphenoid sinus. Sphenoid sinus is located more inside the skull within the sphenoid bone, so it's hard to see on this diagram from the front. Even on the side view, it would be inside structures, not on the lateral sides. And the final one that I wanted to mention is going to be the ethmoid sinuses. So there's a bunch of sinuses within the region of the ethmoid bone, right behind the nose. And I prefer to use the term ethmoid labyrinth to describe that, just because I think it describes a little bit better what's going on here. So these cavities are connected to each other. They look a little bit like a labyrinth-like structure. And so that's a good description of that. So you might ask me, okay, I know these sinuses now. Why do they matter? Why do we have them? Well, there's some agreed and known facts, and there's some proposed theories that are still being kind of looked at. And I think that that's so exciting about the A and B, that there's still a lot to be done. We're far from knowing everything. So first, kind of an argument for having sinuses, which is fairly well agreed by everyone, is that they serve the purpose of reducing the weight of these bony structures that form our skull. Think of the birds. Birds have hollow bones. That's because we are able to reduce some of the mass of the skeleton of theirs, so they're able to fly. So similarly, we want to reduce some of the weight of the skull so that it's not as much to carry up. The second function that I see for the paranasal sinuses is something that we've been able to actually show pretty well, that your voice requires these paranasal sinuses to generate that resonance that we hear and perceive as a human voice when we speak to each other. Now we get to a little bit more of a field where these are more hypotheses than commonly agreed facts. The next explanation could be, or is that it could be that our paranasal sinuses work for shock absorption. And I'll explain what that means. So similar as you have a car, and car has the front where the motor is located, headlights and all that stuff, and then the passenger sits behind that. That design is there for a purpose. Most of the time when we have car accidents, we end up hitting something with the front of the car first. And if you have ever watched an car accident, you'll notice that the front of the car kind of collapses. It goes crumbles up way more than you would expect. Well, that's actually by design. And the idea with that is that rather than coming to a direct stop, that's a very strong impact to the passenger there, let's absorb some of that shock by letting the front of the car to collapse. So similarly, it has been proposed that maybe these paranasal sinuses to a certain extent could absorb this shock, so that if we were to hit our face down to somewhere or face first to somewhere, the impact wouldn't be quite as hard to the brain especially. I'll let you choose whether you think that that's an explanation that makes sense to you, but that is one of the explanations that has been proposed. Finally, we have noticed recently that these paranasal sinuses do have a role in the immune system and functions of the immune system. We don't fully know yet what, but there is something there. So again, anatomy and physiology is a living science in terms of that there's more being found all the time. So the role of the immune system is slightly unclear, but it is something that we're looking to very much. Well, here we see a clinical condition called sinusitis. You remember when it was an itis, it was an inflammation of something. In this case, of course, inflammation of the sinuses. And on this picture CT image that we see on the left-hand side, the maxillary sinus is filled with products from the infection. Few other structures that I want to highlight here. There is an opening at the very posterior part of the nasal cavity, and this opening is known as auditory meatus. So that's a connection from the nasal cavity to your ear, and especially to the inner ear. So sometimes when you drive up a mountain or take a plane and it climbs or descends, you can hear your ears popping. That's because there's a pressure difference between outside your body and the inner ear. And having this connection to the nasal cavity has been speculated that is used to kind of even out that pressure difference. So that would be an explanation when you sometimes, for example, when you travel on a plane, back in the good old days at least, you were given some mints or chewing gum, and we say that helps with this ear thing. The idea is because you swallow, you produce saliva and you swallow that saliva, that helps you to make sure that this difference gets addressed. Well, the next thing that I'm going to talk before we wrap up the nasal cavities role and noses role is that what do we see as a result of this? Why is it so important to have a nasal cavity? Why don't we just breathe through our mouth? Well, first of all, we saw that there was a lot of filtering happening within first at the nostrils, at the nares, and then within the nasal cavity. And a lot of that filtering was thanks to this mucous membrane that we saw, this sticky, wet, thin surface layer that lines all of these parts. Well, this stickiness, the humidity, and the fact that there's a lot of blood vessels that run very close to its surface, so it's able to receive a lot of heat that's transported with the blood. In addition to these conchae that we see from the wall coming, all of these serve a purpose. So in addition to filtering at the level of nares with the hair, that is in the nostril, this sticky surface of the mucous membrane within the nasal cavity is also excellent in grabbing any particles that we don't want us to breathe into the lungs. The other function that we see is that it provides heat to that air. So because there's so many blood vessels running right at the surface of this mucous membrane, we're able to get the air that passes next to it to heat up a little bit. And finally, that being sticky and wet environment surface, we also want to moisten the air that travels from the outside of the body to the rest of the respiratory tract. So all of these functions are made possible by the mucous membrane of the nasal cavity, adaptations of the nasal cavity, in addition to the hair, the conchae, and so on. So we want to filter, heat up, and moisten the air that enters to your body. If you have ever gone out on a cold, dry day, taking a deep breath with your mouth of air, you might start coughing. That's an example when the air entering into your lungs hasn't had a chance to get filtered, warmed up, and moistened. So your lungs react to that this is not ideal. So that's the main reason why the nasal cavity becomes so important for us. We're going to continue a review and next look at the pharynx. So the pharynx is this region that starts posterior to the nasal cavity and continues all the way to the start of the next structure, which is going to be the larynx, so where the trachea and oesophagus divide into two. So we see those points on this diagram quite nicely. In terms of the pharynx, parts of it is shared actually by the digestive and respiratory system, but not all parts. Well, if we look at the pharynx, we can divide pharynx into 3 regions based on location. Most superiorly, the nasopharynx, right behind the nasal cavity, right behind the oral cavity, the oropharynx, and inferiorly, the laryngopharynx, above the larynx, superior to the larynx. Well, larynx then is the next structure that we're going to look at. Larynx is the structure superior to the trachea, and this is dedicated only for the air. This is a combination of muscles, cartilages, and other elastic connective tissues that forms this structure. And what we're going to focus on, we're going to look at various cartilages that form the larynx. So on this diagram, you can see them as these bluish structures, but I've got a little bit more simplified diagram for us that helps us to see what is important about these structures, what you need to know about them. So here on the left-hand side, a view from the front, and on the right-hand side, a side view with half of the larynx cut off, so we can see inside. So let's go through these cartilages that form the larynx. So the first one is going to be the largest of those, are thyroid cartilage. And you can see the thyroid cartilage has actually some good surface anatomy structures for us for identifying it. It has something that's known as a laryngeal prominence. So prominence of the larynx, that is, of course, our Adam's apple, which is more pronounced on the males than it is on the females, but can be seen, felt on both. So if you want to find your thyroid cartilage, go first, find the laryngeal prominence, and from there you can follow to the sides and downwards to see rest of the thyroid cartilage. Well, the term thyroid cartilage also gives us a clue that thyroid is located roughly at this region. So that's thyroid gland. So that's something that we're already familiar from our previous studies. So we do have one thyroid cartilage. Next structure that we're going to look at is located inferior to this thyroid cartilage. So the most inferior, lowermost part of the larynx. And that's known as a cricoid cartilage. Cricoid cartilage is actually the only fully closed, fused, ring-like structure of the larynx, right at the bottom. And again, if you were able to find your laryngeal prominence, find your thyroid cartilage from there, you can follow that as a line and find your cricoid cartilage inferior to that. So I encourage you, if you want to kind of connect these to the real world, a lot of students find it helpful to feel them out on their surface anatomy. Next structure that I want to talk about is going to be our epiglottis. Epiglottis is this flap-like structure located inside the larynx, so we can't see it on this view other than just right from the top. The side view is probably a little better. We see that it is this flap-like cartilage structure that's covered by soft tissue as well. And that is the flap that closes and seals off the trachea, the windpipe, whenever we swallow either liquids or food. Otherwise, at all other times, it is kept open. So that tells us about the importance, the priority of securing the constant air supply to the lungs. That's more important than securing the passage of the food. So we only seal it off when we eat or drink. So that's our larynx. You can actually see on this image an unusually highly protruding epiglottis. So it's peeking out from there. We can see our uvula, our tongue. So normally our epiglottis does not climb that high up. But this was an interesting patient, and I thought that let's snap a picture. Few other structures that I want to discuss next. We actually have two of these neck structures, and these are known as our arytenoid cartilages. So arytenoid cartilages are on both sides, right and left, and they're these triangular shaped structures. And there's a bunch of muscles that connect to our arytenoids and are able to move their angle a little bit. And this is going to be important, the moving of their angle, because it is the vocal cords that are connected right to these arytenoid cartilages. So by them moving, we're able to either tighten or loosen these vocal cords. So we're able to do that change that effect. On top, at the very tip of the arytenoids, we have our corniculates. Corniculates are these tiny tips above the arytenoids. And final one that I think that we need to address, although it is a structure that's usually embedded within the soft tissue of the larynx, so we would not see it unless we do some dissection, is our cuneiform cartilages, one on each side, right and left. And they are, like I said, inside the mucous membrane wall on the lateral sides. So those are our cartilages that make up the larynx. Like I said, there's a bunch of other structures involved, but the cartilages are probably the most important ones for us to be aware of. What is the function of the larynx? Of course, keeping the airway open. We remember that epiglottis was open at all other times than when you are either drinking or eating something. So that is going to be our priority, to keep the airway open. And in addition, this is where the separation for the roots, for the root of the air to the lungs, down the trachea, and route for the food down the esophagus are separated at the level of the larynx. And of course, this is where the vocal folds are also located, and that's where the layman's term for the larynx as a voice box comes from. So you might have heard someone speaking about this structure, referring it as such. Well, I want to mention a couple of words about the vocal cords located in this area. We kind of made a start of it, but let's add a little bit of more detail on that. So here we can see these arytenoid cartilages, one on left, one on right hand side, and the vocal cords coming from them. And these vocal cords, their tightness was manipulated by moving these arytenoid cartilages. You notice that these vocal cords are made of elastic fiber, so there's no direct blood supply, so that's why they look a little pale whitish in color as well. Couple of other terms. The opening between the vocal folds, this space in between, is known as glottis. The proper anatomical and physiological term for that would be rima glottis. But I'm quite happy if you just call it as glottis. Both terms will be perfectly acceptable. So the space between the vocal cords. The other thing that I wanted to mention here is that we actually have two sets of vocal cords that we can end up seeing. More superiorly, we have vestibular folds, which are often known as false vocal cords. That's because they don't really play a role in this voice production, as we are used to thinking of the vocal cords playing a role. The vocal ligaments are located more inferiorly, and they are what we refer as true vocal cords that really do these vibrating edges that we can manipulate to then change the tension on those, and that affects the airflow through past those and allows us to adjust the pitch. There is an instrument in Australia where I spent a lot of time known as didgeridoo, and to play that you actually end up manipulating this glottis of yours. So it's if you're into music, you might want to check that out. There's a lot of videos on YouTube. It's kind of an interesting Aboriginals instrument that has been then adopted and many play, but that is a skill on its own to be able to manipulate your voice box to make the sound from there. In terms of the pathologies and medical terminology. Of course, you can have situations where one needs to perform tracheotomy. This is the incision to the windpipe, to the trachea. In case there's an obstruction somewhere here, high up, that doesn't allow for the air to pass through this area. So we need to secure for the air to come. So how we do this is by making this incision to the trachea, to the windpipe. And this incision, this opening, that making that is known as a tracheotomy. We're gonna then move on to talk about the trachea. Sometimes also you can see in layman's terms referred as windpipe. So that's a other term that you can sometimes see being used. So it is this tube that carries the air from our larynx all the way to the smaller branches of the respiratory tree. It is in length about 4 inches and a diameter about 3/4 of an inch, running, like I said, from the larynx to the mediastinum of the thoracic cavity. Trachea is characterized by being very flexible. And how this is made is that we have these cartilage structures, these C-shaped rings, and I'll talk about the C-shape in a moment, but all you have to think of at this point is that we've got these cartilage structures. They're not directly connected to each other. In between them, we've got a connective tissue that allows a little bit more flexibility. So it allows a little bit more movement for the trachea. And like I said, these cartilage structures that we saw, they are often described as a C-shaped rings. So they're open from the back. So in this diagram that I have here, we're seeing an individual that's looking to the left-hand side. So the eyes and nose are to the left-hand side. So that's the anterior surface. And on the right-hand side, we've got a posterior, the back of the head of this individual. So you can see this C-shaped ring that is part of the trachea. So trachea runs inside there. So why would we have that these rings are not fully fused from the back, from the posterior? Well, that can be explained by us having our oesophagus, the tube carrying the food from the mouth to the stomach, being located right behind it. And oesophagus is most of the time squashed closed, so that's not really an issue. It doesn't take space, and we don't really want to expand it until there's food to pass through. Sometimes you might have swallowed something that has felt a little bit too big to swallow, so that requires for the oesophagus to stretch. So this open back of these C-shaped rings facilitates this, that if there's something large that needs to be passed down the oesophagus, it's able to extend a little bit into the trachea in that way for the material to pass through and pass down there. A couple of other structures that I want to mention. So we see here again our trachea with those cartilage rings, and it divides into 2 branches at the mediastinum. The point where it divides into 2 is known as carina. And that's an important structure. For example, if you look down someone's throat, down the trachea, that's the point where with the camera you can see kind of a sharp tip, or not super sharp, but you can see a tip, and that point is known as carina. Then we have to make a choice. Are we going to the right or left lung? So from there, the trachea divides into 2 main bronchi. And one thing to notice that the trachea is covered all over with this kind of a hair-like microscopic structures. These sweep constantly material upwards in the trachea and respiratory tract. So if there's impurities, some sort of particles that shouldn't have made their way to the lungs, it is these structures that wipe it up to the to the level of the throat so we can swallow it or then cough or spit out. In fact, if you're a smoker, for example, you're giving extra work for these structures. They try to desperately clean your lungs. So that explains why you can sometimes cough, for example, dark matter if you be or dark stuff if you've been breathing a lot of smoke or environment where there's a lot of sand particles, fine sand, and so on. Well, I mentioned that we had carina at this point where the trachea divided into two primary bronchi. Well, let's have a look of these primary bronchi a little bit. What you'll notice is that the right primary bronchi that goes from this point to the right lung is actually coming in a little bit of a steeper angle than the left primary bronchi. This steeper angle is especially relevant if we look at, for example, babies, not babies, but children who have a tendency of putting things to their mouth, sometimes swallowing, sometimes inhaling those into your lungs. So if there's a foreign object that has made its way in this way to the lungs, it's usually found within the right lung because of the steeper angle of the right primary bronchi. The left primary bronchi is not quite as steep in terms of the angle, so it's less likely to find the object there. One more term, I think that we'll see this term again, but I might as well cover it here. The point where the primary bronchi enter into the lungs, penetrate the lungs outermost surface, is known as hilum. And then we have multiple different branches, primary bronchi, secondary bronchi, and so on as we travel down the respiratory tree. We end up with the bronchioles, ducts, and eventually to the level of alveoli. That's the last structure that we'll look. But before we run there, we're going to have a look of the lungs at the larger scale. So lungs fill up the entire thoracic cavity with the exception of the mediastinum. And there's a couple of terms that I want to discuss in terms of there. So you've seen the term apex already earlier on when we talked about the heart and how heart's apex was pointing anteriorly, inferiorly, and left. Apex is a sharp end of a structure. And in this case, the apex of the lungs is located as the most superior part, right deep to the clavicle. So that part is known as apex. Of course, the base of the lungs is the inferior-most surface resting on the diaphragm. And one thing that I wanted to highlight here is that look at those lungs and how their base covers, especially on the lateral side, much further down than students often think. So lungs do actually go quite far down on the lateral sides. So something to bear in mind there. And finally, the term hilum, we saw that already before, the site of the entry of the primary bronchi to the lungs after they have divided at the spot of the carina. So a couple of other terms, you can look at those yourself, root and coastal surface. But what I want us to focus now is the lungs and the lobes and fissures of the lungs. And let's start by just making the difference between right and left lung. And what we see is that the left lung is smaller. And this size difference is because you remember we had a heart, and heart was located a little bit more on the left side of the thoracic cavity or from the midline. So it takes some of this space from the left lung, making it smaller. And it is the cardiac notch, the term that we call what we see then on the left lung as this curvature that facilitates the space for the heart. So that causes the left lung to be smaller, and we end up finding less structures there. Well, let's look at the different lobes. And to draw those lobes or locate them, let's look at the fissures first. So you can notice on the right lung, we've got two fissures. The first one runs a little bit horizontally. If you have a little bit of imagination, you can see that at large, it's kind of a flat like our horizon. So that's the horizontal fissure. The other one is oblique fissure because it doesn't quite fit into any of those planes that we've seen before. And these fissures help us to identify three different lobes of the lungs: superior, middle, and inferior lobe. And as we look at the left-hand side, we notice that there's going to be only one fissure. This is going to be the oblique fissure, and we're left with the superior and inferior lobe. So this is the general, what we find most of the time. Of course, there's some individual variation, and it is more common than you probably think to end up finding different number of lobes in individuals. But this is what's in most individuals, and this is what we're going to learn. So we can add more of that individual variation once you get further in your studies. Other thing to notice is that the lung is surrounded by a double layer of membrane, the inner layer known as visceral layer and the outer parietal. Between them we have fluid, and this fluid allows these layers to slide easily past each other, cushions the lungs a little bit, and most importantly generates the negative pressure within the thoracic cavity around the lungs, so that the lungs are pulled constantly open as large as they can be. And we'll see consequence if this gets compromised, what happens then. I think that on here, I simply wanted to discuss about the fact that as we are moving down the respiratory tree, we end up seeing more cartilage at the superior portions, and we also see that there's less smooth muscle at these superior portions. But as we move down the respiratory tree, the amount of cartilage decreases and the amount of smooth muscle increases. I'm going to say a couple of words about the alveoli as well. Alveoli are these very end pieces and parts of the of our respiratory tree, kind of these sac-like structures with very thin wall, clusters of them. And there's about 300 million of those in one individual, and they actually make up most of the lung volume. And these are especially important structures because this is the site where the gas exchange happens. So here we have the wall of an alveolar cell. So the lung would be on the left side, and we also see a small capillary of the pulmonary circulation right in connection right next to this alveolar wall. And this facilitates that it's easy to exchange these materials for the oxygen to enter to the blood and for the carbon dioxide to leave the blood and enter into the alveoli and lungs, so we can get rid of that by breathing it out. So it is the alveoli that's the site of the gas exchange. Well, let's look at the physiology a little bit. So we have respiration can be divided into a few different phases. I've presented to you 4. There's some debate if this is the best model, but I think it's the best model we have available. So we're going to use that as we go through this structure here. So the first stage of respiration is known as pulmonary ventilation. It refers only to this very first event where we carry air from outside of the body to the lungs and all the way to the alveoli. So I've drawn a single alveoli on this diagram. So pulmonary ventilation, simply carrying the air from outside the body to the lungs and to the alveoli of the lungs. And of course, we have 2 stages of that: breathing in inhalation and breathing out expiration. Well, the next stage is going to be the external gas exchange. So it happens here where gases are moved between the alveoli and the capillaries of the pulmonary circulation. So you remember pulmonary circulation was this loop from the heart to the lungs and back to the heart, which brought deoxygenated blood, and it came back as richly oxygenated. So it is these capillaries that run right next to the alveoli, this short distance between the two, like we saw just earlier on here, that facilitates this exchange of the gases. And the exchange of the gases is done based on the partial pressure difference. So materials move from high concentration to the low concentration. And we'll talk about that in a little bit more detail in a bit. The following step after that is going to be the gas transport. So our systemic circulation, when we get the blood that's rich in oxygen coming from the lungs, transported to our tissues in the body, and from the tissues in the body, we pick up the carbon dioxide, leave the oxygen there, and carry the carbon dioxide rich blood to the heart as part of the systemic circulation. So that's our gas transport phase of the respiration, phases of respiration. And final stage, probably not surprisingly to you, is going to be the internal gas exchange. Since we had external, there's going to be internal as well. And this is where materials, the gases, are exchanged between the capillaries within the systemic circuit and the cells or other structures that might need the oxygen, pick up oxygen and get rid of their carbon dioxide. So that's the stage of the internal gas exchange. And of course, remember that it is not only just bringing the oxygen in, it is also getting rid of the carbon dioxide, breathing that out. So these stages go back and forth. So we can go 1, 2, 3, 4, and then again 4, 3, 2, 1. So the order of them, it just describes it one way. The order is opposite to get the gases out from the body. This was what I referred earlier on, that if we have a puncture on the pleural cavity that surrounds the lungs, well, as a result of that, this puncture here will break this negative pressure that we have within the pleural cavity, keeping the lungs extended. Now this pressure, negative pressure, is lost, and the lung will collapse. So this would not be a good thing for us to happen. I want to discuss next about breathing processes. So breathing in, which actually takes effort. So if you sit there and just relax, you'll notice that no air is flowing into your lungs. This takes effort. So this is active process. So we need to recruit 2 muscles to do breathing. These muscles are going to be external intercostal muscles, so the muscles between the ribs and the most external ones of those, and diaphragm. It is the diaphragm that's your main respiratory muscle. And I like asking that in the exam, just as a FUY, in case you want to make a note of that. So diaphragm is the main respiratory muscle. What external intercostals and diaphragm do, they increase the volume, the size of your thoracic cavity. So your thoracic cavity grows bigger and bigger and bigger. And this is how we're able to accomplish this difference in the volumes and the amount of molecules within this space between outside the body and within the thoracic cavity. So when you breathe in, you extend your thoracic cavity. As a result, these molecules that were earlier on in the air within your lung, now that the volume of this space is larger, to cover the entire space, they have to be located further distance from each other than they were before. So now we develop inside the lungs low pressure, whereas the outside of the body is going to become high pressure for these gases. And you remember that gases always move from high pressure to the low pressure. So if outside of the body is high, air flows in to the lungs and chest cavity as you extend the size of it. So what we'll see, this diaphragm, which is usually kind of dome-shaped muscle at the bottom, that flattens out, making the chest cavity bigger. And like I said, the external intercostals lift up the chest cavity and make it larger. Well, expiration, so breathing out. If you take a deep breath and then fully relax, you'll notice that some air flows out. So there is normal elastic recoil. Just naturally, your ribs kind of return to their shape and return the shape of the chest to the shape that we're used to. This happens passively. You don't need to invest any energy on that. And what we see now, the thoracic cavity and lung space as well becomes smaller. It decreases in size. So let's look at what we just looked before. So now here we have our lungs and we have air molecules within there. If now we make it smaller, the same amount of molecules remains there, but they are now packed much closer to each other. So as a result, we develop high pressure inside the lungs and low pressure outside the lungs. And again, materials traveled from high pressure to the low pressure, so air flows out as we shrink the size of the thoracic cavity and the lungs. Now air flows passively out as this shrinking is a passive process. It happens just even without effort. Of course, you can breathe out forced exhalation, forced expiration. So now you make your chest even smaller than it normally would be. You make your thoracic cavity even smaller. So you recruit oblique abdominal muscles, transverse abdominal muscles, and internal intercostal muscles to make your chest cavity, make your lungs as small as possible. And as you make them as small as possible, we remember again, we end up noticing that for the same amount of the air that used to be in our lungs, now it's packed into smaller volume. So there's going to be high pressure within the lungs. And air flowed from the areas of high pressure to the areas of low pressure, so out from the lungs. And we are now doing active process since we're recruiting these muscles and going beyond what would be the normal size of the lungs during air exhalation. There's a bunch of other air movements that can take place that are not really involved with the breathing, coughing, sneezing, crying, laughing, hiccups, yawning. All of those are air movements, but they don't necessarily relate immediately directly to the respiratory functions. A couple of other terms, surfactant. I often think of that as helping with the diffusion of the gases and also keeping the alveoli from collapsing. Compliance, I think of this as a measure of the stretch that the lung has. How do we measure then lungs and assess them? Well, you might have seen this tool that we see on the right-hand side. That's our spirometer, just a plasticky tool that we use to measure the volumes of the patient's lungs. And there are more sophisticated versions that are connected to computing resources and electronics. So, but the basic principle is the same on both. So what we can measure with these is the respiratory volumes. And by using respiratory volumes, we're then able to calculate respiratory capacities. So let's have a look at the respiratory volumes first. Well, you'll notice this wiggly line. The area that I've highlighted on the yellow is the normal breathing. You breathe air in and you breathe air out. But if you take a maximal breath of air in, you get this high peak that goes up on this graph. And if you do the forced exhalation, so breathe out air as much as you can, you get this peak going down. So if you compare this, what's normally happening, and these extremes that we can do, you'll notice that normally we don't use nearly all of our lungs volume that are possible. We only do use very little of that, but we have that extra that we can use if we need. So let's add some terms for different volumes that we can measure. So I mentioned initially this area of orange, which was our normal breathing, how much we normally breathe in, how much we normally breathe out during the normal breathing. That's known as our tidal volume. And the key here is, like I said, that we're not breathing in more than normally. We're not breathing out more than normally. The normal amounts make up our tidal volume. And that's about 500 milliliters on a typical individual. We can also breathe in more than we would normally breathe by doing this purposeful extra breathing in. This is known as our inspiratory reserve volume. So this is when we do the breathing in beyond the normal. We apply some force into this, and this is typically somewhere in the range. There's quite a lot of variation at 2100 to 3200 milliliters. So quite a lot more than what our tidal volume was. And of course, we have the same for breathing out. We can breathe more air out than we normally would. So this is again forced, putting in extra effort to breathing out. Our expiratory reserve volume, typically it is somewhere around 1,000 and 1,200 milliliters. So not quite as much as our inspiratory reserve volume, but still quite a bit. And then we have a portion of the lungs where the amount of air, which represents the amount of air that's always present in the lungs and respiratory tree. So some is needed to keep those lungs open. Some remains in the respiratory tree. We can never fully empty it. And this is known as a residual volume. So now that we've gone through these different volumes, we can use them to calculate now various different capacities that we see on an individual. So let's have a look at those. So first of all, we're going to look at the inspiratory capacity. This is the sum of adding our tidal volume and inspiratory reserve volume. So as much as we can breathe in plus the normal breathing in gives us our inspiratory capacity, the capacity of how much we can possibly breathe in. Well, the opposite, then for if we look at functional residual capacity, that's our residual volume, so the volume of the air that always remains in the lungs, in the respiratory tree, plus the expiratory reserve volume. So they make up our functional residual capacity. We also can have vital capacity, where we add together the tidal volume, inspiratory reserve volume, and expiratory reserve volume. So really the amount of air that can be from max to the minimum in the lungs. And finally, total lung capacity is adding all of those previous ones together. So we include the tidal volume, inspiratory reserve volume, expiratory reserve volume, and residual volume to calculate the total lung capacity. Another thing that I want to mention, there is always some air that remains in the passageways. This is known as anatomical dead space. And there's always some air that kind of remains in the parts of the lungs, the alveoli, that are no longer functional, that are kind of waste part that no longer serves its purpose. This is the alveolar dead space. So total dead space is the sum of these two. In terms of the gas exchange, do remember that external respiration was this exchange of gases between the lungs, the alveoli of the lungs, and the blood, the capillaries of the pulmonary circulation. Whereas the internal respiration was the exchange of materials between the capillaries of the systemic circulation and the tissues of the body. And remember that the gases traveled always from high concentration to low concentration. So it's the partial pressure difference between different parts of the where the exchange happens that drives the movement of the gases. And this is typically measured as millimeters of mercury. So we could go into more details of looking at different laws, but let's keep it at that level. So let's see what was happening here. Here we are seeing the exchange of the gases between the alveoli of the lungs. So that's this part here. And the capillary of the pulmonary circulation, this vessel running here. Once the blood comes in the pulmonary circulation, it has about 40 milligrams of mercury of oxygen concentration, whereas the oxygen concentration within the alveoli is about 100 millimeters of mercury. So materials move from high concentration to low, so oxygen moves from the alveoli of the lung into the capillary of the pulmonary circulation. Conversely, if we look at the concentration of carbon dioxide in the blood coming back from the pulmonary circulation to these capillaries, about 45 millimeters of mercury, whereas the concentration within the alveoli is about 40 millimeters of mercury. So now gases travel from these capillaries of the pulmonary circulation to the alveoli of the lungs. So from areas of high to the low. Well, let's look at these events in the systemic circulation between the capillaries of the systemic circulation and the tissues of the body. Well, blood coming to these areas, of course, it's going to still have that 100 millimeters of mercury for the partial pressure of oxygen, whereas partial pressure of oxygen within the cells is only about 40 millimeters of mercury. So again, we move from high pressure to low pressure. So from the capillaries to the cells is where the oxygens go, whereas carbon dioxide, the concentration in the blood, in the systemic circulation, the blood that comes from the heart is about 40 millimeters of mercury, whereas it is about 45 millimeters of mercury within the tissues. So carbon dioxide travels again according to the concentration gradient, so from the cells to the capillary blood of the systemic circulation. How are the gases transported in the body? Well, about 98%, so the majority of oxygen is bound to the hemoglobin of the blood, only about 2% bound to the plasma, whereas carbon dioxide, about 3/4 are transported as bicarb, 15% bound to the hemoglobin, and 10% to the bound to the plasma. So we see again this familiar diagram or familiar equation that we saw before. This was the cellular respiration where we took sugars and oxygen and those were converted to energy, so ATP, and as a waste product we were left with carbon dioxide and water. So we got that oxygen into this process from the respiratory system, and the sugars, of course, from the digestive system. So we are taking both of those elements into the body, so we can make this ATP, the energy. And we're left with the carbon dioxide and water as a waste product, like I mentioned. So let's move the carbon dioxide and water to this second equation that we see at the bottom. Well, we need to get rid of the carbon dioxide and water somehow. How we get rid of it? We convert them into carbonic acid, which then can then be converted further to bicarbonates and hydrogens. Well, we get rid of the bicarbonates through the respiration, so we can breathe those out, and hydrogen ions, we get rid of them through the urinary system when we urinate. And that will be a chapter that we look at a little later on this course. So are there effects that the altitude has on the oxygen content that we see in the air? Well, there is. So like you remember, let's start with the sea level. The oxygen molecules in the air are some distance apart from each other. And that's a normal state where we normally breathe, living. That's enough oxygen for us to perform and live quite happily at the sea level. However, if we go to high altitude, we notice that there's less oxygen atoms in the same volume of air, so breathing becomes a little harder. If you have been living or climbing a lot, you'll notice that it feels like doing everything is a little laborsome at the high altitudes. That's because there's less oxygen available. So we're not quite as efficient as we are used to be. Well, some athletes find this beneficial before important competition. They go and train at the high altitude camps, where their body has to, over time, adjust to the low amount of oxygen in the air, and it becomes much more efficient of getting more oxygen into your tissues. So as a result, once they return to the sea level, they end up outperforming their peers who have not been training at high altitude because their body hasn't done this adjustment to the high altitude, and they just perform the normal metabolism of normal use of oxygen from the air, whereas these ones who trained at the high altitude are much more efficient because their body kind of went on the overdrive of adjusting to this challenge. Of course, we have a group of people who train at the high or who live at the high altitudes, and they have adjusted as a population to those, and we noticed when we studied their physiology that their body is perfectly adjusted to these high altitude conditions. Well, what happens if you want to have more detail? You can look at the full lecture video, but to give you a short summary, high altitude, when there's little oxygen, you remember we started producing erythropoietin. That was a hormone that your body produced. And erythropoietin then resulted, if you were in high altitude, lack of oxygen, for you to produce more red blood cells and transport the oxygen more efficiently to your tissues, such as your muscles. Well, that can be produced naturally if you're training on those high altitude camps that we discussed before competition. But you can also take injections of this hormone, erythropoietin, and that would trigger the same sort of a response. But of course, that would be considered as cheating, doing doping. Where the respiration is controlled, well, from the brainstem, from the structures of medulla and pons. You remember the medulla oblongata and pons as some of our most primary, most earliest structures that developed of the brain, and some of the most earliest structures that develop not only in an evolutionary sense, but on individuals' development as well. So breathing is very important. It develops early, it's preserved on brainstem, in kind of protected as much as can, whereas the other parts of the brain are a little bit more sensitive and at the risk. So how much do you normally breathe? How many breaths per minute? Well, we all have different variation based on various factors, but typically we're looking at a range of 12 to 20 breaths per minute in an average individual. You will notice the newborns, and because I like cats, I always include facts about cats. Not only newborns, but also cats, their breathing is faster. So if you look at the newborn and they seem to be breathing a lot, do not freak out. That's normal. Same goes for the cats. And also their breathing is a little bit less regular. So it's a little irregular in comparison to what you see on an adult human. Of course, a condition that we can relate and consider here is hyperventilation. This is when one breathes really deep and really fast. So you go something like, well, if you keep doing that for a long time, you actually exceed the body's need to remove carbon dioxide, and that will cause us issues. So your blood carbon dioxide levels drop, and as a result, you can get dizzy, even faint. So we need to avoid that from happening. So if someone's hyperventilating in the past, I think we gave them a paper bag where they were breathing then so that they would get some of that CO2 that they were putting out breath back into their lungs. If someone's hyperventilating, I would encourage you to try to calm them down, tell them that if they don't slow down, they might get worse off, they might get dizzy. So try to breathe in that case a little slower, not as fast. And you don't need to have as deep breaths either. Well, covers all that I had in terms of the study material from this chapter. And I'm just going to jump to to the PowerPoint that I had earlier on to just show a couple of last points that I want to emphasize. Let's get that up. And I know that that was a lot of information in a quick period of time. So like I said, there is a full lecture series of lecture videos available on your Canvas course. And I'm trying to keep this even a little bit more condensed than the morning's video. So we have covered now the key points from the respiratory system chapter. The last things for me to remind is that we do have tutoring services available. Everything's restored. They work now. You don't need to make an appointment. You just go to that link and it's a virtual tutoring service. You can choose the tutor you prefer, or if they're available, they'll meet you immediately. If they're not, you can just wait for them to become available. Of course, I'm also available for you. I've done quite a bit recently of going through material with students on one-on-one basis. So do not hesitate to let me know if you feel that that would be beneficial. I am also available in addition to the using the tutors of the institution. Finally, last thing for me to say is that if you have any questions, I really encourage you to use the Canvas messaging system. I know I have few messages that I'm yet to answer there because I only got them now. So I will answer them to ASAP. But any questions, whether it's a technical question or material related question, let me know. I'll try to answer. If you want to have one-on-one session, you just let me know that you're looking for that. We'll find a time that works for both. And on that note, I do think that we have covered everything. Like I said, the systems outages are now done and dealt with. There's still a little bit of things that I promised to fill you in terms of the gaps of some things that we're doing changes. I think most of the due dates I was already able to adjust. There might be few that I look to do a little later, but we've gotten over that hurdle. We're at the good place. And I do really want to thank you for your patience as we've been dealing with this. I can ensure that it's been as challenging and as confusing time for everyone than just students in the college. So on that note, this is it from me for today. I appreciate you joining me. And I will make this recording available in just a due course on the Canvas course. Thanks again. Let's make it a good week. And remember, I'm here to help you. All you have to do is reach out.